All right, welcome to part 2 of chapter 9, where we're going to look at protein synthesis and then see some examples of mutations. Ricin. Have you heard of ricin before? Ricin is a toxin, very, very deadly poison that used to be especially the poison of choice by assassins. It's banned as a weapon under the Geneva Protocol. People still use it. Every now and then you'll hear about somebody purifying ricin from castor oil. That's where you find it, in the castor oil plant. And then they'll try to use it to poison somebody they want to poison. Ricin targets ribosomes. And what it does is it inactivates the ribosome. And if you inactivate ribosomes, you can't build proteins. Think of all the things we've learned that proteins do for cells. You know, whether they're catalysts and control biochemical reactions, or they're contractile, or they're transport, or energy storage. There's so many roles of proteins. And when you can't make proteins, cells aren't going to be able to replicate, cells aren't going to be able to maintain, and you're going to die. And so that's why we want to be careful with who gets their hands on ricin. Remember, DNA stores hereditary information, and RNA is used to build a protein. There are 3 types of RNA. There's messenger RNA, transfer RNA, and ribosomal RNA. Ribosomal RNA builds the ribosome. Transfer RNA carries amino acids to the ribosome from the cytoplasm, and messenger RNA is a way to take information from the DNA to the ribosome. See, the ribosome's made of RNA, and the information, hereditary information, is on the chromosome in DNA. The ribosome can't read DNA, so we've got to rewrite the DNA into a form of RNA that can then go to the ribosome to be used to make a protein. And a gene is a section of the DNA that codes for a product. Okay, so if we want to look at if we're going to build a protein, if we build a protein, then we're expressing the gene. We're using the gene to build a protein. If we're not building a protein, then we're repressing the gene. If you think about it, if you take a living cell in your nose, it has your entire genome. It has the information to build any protein, any structure that you have in your body, right? So there are genes in the cells of the nose to build an eye, or to build a hand or an arm or a foot, to build a heart, a kidney. All that information is there, and yet you're not building those things in the cells of your nose. So most of the time, genes are repressed. They're only expressed when we want to build a protein because we need it to either replicate, grow, maintain, or repair. So the process of taking DNA to mRNA is called transcription. And what we're going to do is first enzymes are going to open up the DNA and expose the gene. Remember, the histones identify where various genes are. The gyrase and helicase unwind the helix, open it up, expose the gene. And now, much like DNA replication, we're going to use the gene on the DNA as a template to build but this time to build mRNA. And the difference is we're going to use RNA nucleotides instead of DNA nucleotides, which means the sugar's different. Ribose instead of deoxyribose. We don't have thymine in RNA. Instead, we have a pyrimidine called uracil, a single ring structure. Okay, and then there are 3 steps to mRNA transcription. Initiation, where we start the process. Elongation, where we're adding RNA nucleotides to the growing mRNA transcript. And then finally termination, when we've gotten to the end of the gene and we need to release that piece of mRNA. Okay, so here's our double-stranded DNA, and somewhere in front of a gene that we're going to copy is something called a promoter. And when that promoter gets activated, it's going to cause enzymes to unwind and open and create a replication fork. And then RNA polymerase is going to start following this and building RNA complementary to the DNA. As soon as we get past an area, this is going to wind right back up again. Okay, and so we just keep following the replication fork until we get to the termination sequence. And now we've got a piece of mRNA complementary to the DNA. Okay? And this is showing that process. right? Here's our RNA transcript. We're reading the gene 3' to 5', so we're building the RNA transcript 5' to 3', and you can see, okay, G complementary to C. We don't have thymine in RNA, so we use uracil across from every A. Okay? We still put an A across from a T, put a C across from a G across from a C. But remember, these are RNA nucleotides, not DNA nucleotides. Once we hit the termination sequence, this RNA transcript is released. Now, there are some proteins in your body and your cells that you need a lot of. And we can very quickly make a lot of a particular protein by making more than one mRNA transcript while we've got the gene open and exposed, and by using that transcript to go through multiple ribosomes at the same time, right? And so here you can see that here we're copying the mRNA, and as soon as it's being copied, even before it's detached at termination, it could be running through ribosomes. This ribosome it went through first. This ribosome is just starting to go through. So the protein will be shortest here. Here the protein will be more developed. And the other, most developed, a little bit less, a little bit less, a little bit less, a little bit less, and finally least developed. So this is how you can make a lot of a particular protein in a hurry. RNA, before the mRNA leaves the nucleus in eukaryotic cells, it has to be modified. And what we're going to do is we're going to splice it. DNA has exons and introns that are present in the gene. Exon doesn't mean outside and intron inside. X stands for expressible. So when you use the exons, you build the protein. The exons can be expressed to build a protein. Introns are intervening sequences. And we're not really sure what they're from. Some we know are viral in origin. There are viruses that when they infect you, they'll put their DNA into your DNA, and it can lie there dormant for a while. And over the time that humans have been on planet Earth, we've accumulated a fair amount of viral DNA in our chromosomes. So part of that is introns. Some of it we think might be regulatory. And as time goes on, it seems every year some scientist somewhere finds out that what we thought was useless DNA actually has a role. And then other scientists think introns may be part of genes that we don't need anymore, that were part of, coded for a protein that we used to need when we were in a different form that we don't need anymore. Once we've spliced out the introns and put the exons together with DNA ligase or RNA ligase, then we've got to add a cap. And a cap is kind of like the cover on a textbook. I mean, the truth is, you don't really need to read the information on the cover once you've looked at the cover one time and know what's inside. Okay, what the cover really does is it protects the nucleotides inside from being damaged, or pages inside from being damaged in a book. The cap protects the nucleotides inside the message from being damaged. And then on the other end, we're going to have what's called a poly-A tail, which is just a whole bunch of adenine nucleotides added to the end. That indicates, which end of the transcript, is the tail end and which is the cap, because the ribosome needs to know that. So here's an example. Here's our gene, our DNA, or our DNA was up here. And when we transcribe RNA from the DNA, we transcribe the introns and the exons. But the introns don't code for the protein. The exons do. And so we're going to clip out the introns, and then we're going to splice together the exons. We're going to leave a little bit of untranslated region, which is part of the cap, and another part here that's part of the poly-A tail. So these are all adenines. These are all just extra nucleotides, but not in the sequence AUG, and that'll make sense in a little bit. Okay, once we've put a cap and a tail on and spliced together the exons, now this transcript is ready to leave the nucleus and go find a ribosome. In translation, first let's look again at the roles of the different RNAs. Okay, so mRNA is the messenger RNA that's complementary to the DNA, that's bringing the instructions to the ribosome, telling it which amino acids need to be added to the polypeptide chain in sequence. And you see here our growing protein chain where we're adding one amino acid at a time. Okay, we need so the ribosome is made of ribosomal RNA, and then transfer RNA is responsible for attaching to an amino acid and then having another site that's complementary to part of the transcript so that we put the correct amino acid in the correct order. And we'll see how this works in a minute. Okay. Now, there are 20 different amino acids that all things use to build proteins. But there are four different letters, 4 different bases possible in the RNA. All right. Well, I can't let every nucleotide in the RNA represent an amino acid because there's only 4 different RNA nucleotides and there are 20 different amino acids. All right. If I let every 2 nucleotides represent amino acids like AA, AU, AC, AG, and so on, 4 times 4 in the first position, 4 in the second, is only 16 different combinations. Still not enough to have a unique combination for each amino acid. But if I let it be 3 nucleotides, 4 in the first position, 4 possible in the second, 4 in the third, 4 times 4 times 4 is 64. And now I have enough different 3 nucleotide segments to represent all 20 amino acids. These 3 nucleotide segments are called a codon. And codons have a number of roles, but it's the order of nucleotides in the codon that identifies which amino acid that codon specifies, and it's the order of codons that determines the order of amino acids in the protein. So if we put all this on a table where, here's our first letter, here's our second letter, and then finally the third letter, these are the 64 possible codons that we could get on a piece of messenger RNA. Now, when you see the 3-letter abbreviation, PHE stands for phenylalanine, LEU for leucine, ILE for isoleucine, MET for methionine, valine, and so on. These are the amino acids. You'll notice that there are 3 codons that code for stopping. This is how the ribosome knows when it's finished making the protein, is that a stop codon slides into position on the ribosome, and that terminates translating the mRNA into a protein. There's also the AUG codon, which codes for methionine. AUG is also called the start codon because it's going to be the first codon that initiates translation. Remember, there's a cap in front of AUG of just random nucleotides, but not in AUG order. Okay, so the ribosome's reading through the cap, and it isn't until it hits the first AUG that it starts translating, putting together a polypeptide chain and start building the protein. Notice that many of the amino acids have multiple, leucine. There are 1, 2, 3, 4, 5, 6 different codons that code for the amino acid leucine. There are 2 for phenylalanine. There are 1, 2, 3, 4, 5, 6 for serine. Okay, this is called the redundancy in the code. And what it does is it protects against mutations, and we'll see this later on. So this is the genetic code. Now, some of the codons coded for amino acids. We call those sense codons because they're coding for an amino acid. They have a sense. A nonsense codon is one of the 3 that code for stopping. Because not nonsense because they're silly, nonsense because they don't code for amino acid. And of course, there's the start codon that initiates translation and codes for methionine. Now, you can have more than one AUG. You can have more than one methionine in a polypeptide chain, but you're going to start with a methionine because the start codon is AUG. Transfer RNA that carries the amino acid from the cytoplasm to the ribosome has 2 important parts. The one part is the anticodon, and this is where we have a complementary sequence of nucleotide bases complementary to the codon. So if the anticodon is GAA, then this is complementary to the codon CUU. Okay? And then the acceptor stem is where we bind the amino acid. And there's a bunch of proteins that function in this too. Okay? So the amino acid is here, and this particular transfer RNA will only bind to one amino acid. And when it brings that amino acid to the ribosome and then is released, it has to bind to another of the same type of amino acid to bring that. So you have lots and lots of different transfer RNAs in the cytoplasm of your cells. Some empty, looking for their amino acid so they can carry it to a ribosome. Some already attached, just waiting for a ribosome that needs them to bring in that amino acid. Some already attached to the ribosome and holding the amino acid in position to be added to the protein. Okay, and this is showing you that ribosome structure, there's a small subunit and a large subunit, and most of the time, these two subunits are separated from each other. They don't come together and become intact until there's an mRNA message that's between them. Okay, so again, transcription was taking DNA and turning it into messenger RNA. Translation is taking that messenger RNA at the ribosome and turning it into a protein. And it starts with initiation. In initiation, the small subunit is reading through the cap until it gets to the first start codon. And then it's going to bind the mRNA, and then the large subunit is going to attach to. They're going to sandwich the messenger RNA in between them. There are two sites on the ribosome. There's an A site and the P site. The A site is where we've exposed the codon, and we're waiting for the tRNA to come in with the complementary anticodon, briefly bind, and with it comes its amino acid. And then we're going to translocate. Once it attaches, we're going to move that tRNA with its amino acid to the P site while we expose the next codon in the A site. P stands for polypeptide, or the peptidyl site. That's where we're holding the growing polypeptide chain. And now the second tRNA comes in and brings its amino acid and attaches complementary to the second codon. And then the large subunit is going to catalyze a peptide bond that links the two amino acids together. That first tRNA is now empty. It gets kicked off. And now the second tRNA that has its amino acid connected to the first amino acid shifts and translocates to the P site. And we just keep translocating over and over and over until we hit a stop codon. And when we hit a stop codon, the polypeptide chain is released to bend and fold or joined with another polypeptide chain to be a quaternary structure. The mRNA is released, and the two parts of the ribosome separate. So if we're looking at that, here's our small subunit, here's our large subunit, right? We've got the A site, we've got the P site, okay? And so now we've exposed a codon. We've already initiated. We've gone through 1, 2, 3, 4, 5, 6, 7, 8, 9 translocations. We've brought in tRNA with the correct anticodon that's complementary to this codon. With it comes its amino acid. Once it's anchored, the large subunit takes this polypeptide chain, attaches it to here. Then this is going to translocate. We're going to expose the next codon and repeat the process. And we just keep repeating that process until we hit a stop codon. Okay. Now remember, a mutation was a change in the nucleotide sequence of the DNA. Now, this can be harmful. In fact, this is how a lot of cancers are formed. But it could also be helpful. What if the change in the nucleotide sequence ends up creating a protein that's a little bit better than the protein was before, that makes the organism more competitive, that makes it better able to use nutrients or to utilize the ecosystem? Okay? So mutations can be helpful. They can be harmful. And they may not change anything because it's possible. Remember the redundancy of the code. If you have multiple codons coding for the same amino acid, it's possible to change the codon but still code for the same amino acid. Okay? A base substitution, also called a point mutation, is when we just change a single base. Right? So here's our DNA, TTC, TTT, ATC, TCC, TGC. Okay. No. Let me rephrase this. Here's our normal DNA without a mutation, TTC, which is going to code for AAG on the mRNA, which codes for the amino acid lysine. Now, if we change the DNA, so now instead of a C, we put a T here. Now we get a different codon, AAG, AAA. Okay, so we've changed the code, but the codon codes for the same amino acid. AAA and AAG both code for lysine. So we've changed the DNA, but we're not going to change the protein. This is called a silent mutation because it doesn't change anything. If we change the DNA, if we change the DNA so that instead of a normal codon, we get a premature stop codon, then we stop translating early and we truncate or shorten our protein. And if we truncate it too much, it might not be functional at all. Okay? If we change the DNA so that we get a different codon that codes for a different amino acid, okay, now we have a missense mutation. A missense because we've changed the sense of the codon so it codes for a different amino acid. Okay, so a base substitution or point mutation, there are 3 possible outcomes by changing the DNA in one spot. Either a silent mutation where we change the DNA, we change the mRNA, but we still code for the same amino acid. No change. A nonsense mutation where we change the DNA, change the codon in the mRNA, and now it codes for stop, and we don't make the complete protein. It's short, truncated. Or a missense mutation where we change the DNA, change the codon in the mRNA, but now we're coding for a different amino acid than what we had before. Okay? A different type of mutation is a frame shift mutation. Okay, in a frame shift mutation, remember we're reading with the ribosome 3 nucleotides at a time. Okay, that's our reading frame. Okay, that's our reading frame. Well, what if we have a deletion or an insertion? Okay, so here we had a T in this position and we lost it. And so now instead of AAG, TCA, CTC, GTA, the AAG is unaffected, but every codon after this is changed and could be coding for a different amino acid or a nonsense codon, or it could still be a silent mutation. But you change every codon after the insertion or deletion. Okay, and so what that means is you have the greatest chance of creating a vastly different protein based on a frame shift mutation, losing or gaining a nucleotide on the mRNA. Okay. Now, genes are controlled in your body. You don't want to build a protein that you don't need. All right. If you go shopping or you drive to the college for a face-to-face class, okay, and like when you come to lab, right? Do you leave your car running in the parking lot? Of course not. It's a waste of energy. It's a waste of gas, right? You shut it off and wait till you need it to turn it back on. Same thing is true in your body. You don't want to build proteins that you don't need. When that's occurring, that's that symptom unexplained weight loss that you hear said very fast in tiny letters at the bottom of drug ads. Okay? This is a situation, something's wrong. Your body's wasting energy. Healthy, productive cells don't waste energy. And so we want to be able to control which genes are expressed and which genes are repressed. Okay? Remember, most cells are specialized and are only going to express the genes necessary to be that kind of cell in whatever tissue or organ that cell is a part of. Okay? Now, how we often control gene expression, remember that promoter that was on the DNA ahead of the gene? Well, oftentimes there's a molecule that has to bind to that promoter to either speed up, slow down, or prevent transcription. And that usually it's a protein and it's coded for by something called a transcription factor. Somewhere on the chromosome or maybe even on another chromosome is going to be a gene that codes for a protein that when it's produced is either going to bind to the promoter to start transcription, speed it up. It's going to bind to the promoter to slow it down or prevent transcription. Okay, but the bottom line is that your gene expression of your genes is controlled. It's not designed to happen when your cells don't need the production of a product. Epigenetics are chromosomal modifications, and these can suppress transcription. They don't change the DNA sequence. Instead, what happens is methyl groups are added to the histones. And when you methylate the histones or direct methylation of the nucleotides, this can suppress transcription because now the enzymes that are going to transcribe the DNA, the methyl groups are in the way and it can't get past them. And there are environmental factors like the chemicals in cigarette smoke that can add more methyl groups to your DNA. Okay, if your parents' chromosomes are methylated, typically when the sperm and the egg come together and fuse to form the zygote, the chromosomes are reset and methyl groups are removed, but sometimes they're not. And so some methylations can be passed on to the offspring. And of course, this could lead to the inability to produce certain proteins. It turns out boys are affected by lifestyles of individuals in the father's line, girls by individuals in the mother's line. That's what we found. And these heritable changes in the gene expression, they're not due to changes in the underlying DNA sequence. We call this epigenetics. And there are people who study this in great detail. Okay, so gene expression is regulated. It begins with transcription, and then mRNA processing, and then translation. Okay, and then we can also post-translational modification of proteins, folding the proteins, adding other molecules to the proteins so that we get a functional protein. So if we go back to the very beginning, we talked about ricin as inactivating your ribosomes. Okay, a ribosome inactivating protein. Ricin's a RIP. Okay, they have one polypeptide chain, binds tightly to carbohydrates on plasma membranes, and then that helps them get inside the cell. Once inside, a second polypeptide inactivates the ribosomes and the cell quickly dies. There are other ribosome inactivating proteins. The toxins from Shigella bacteria and E. coli bacteria behave like this too, only they're not quite as toxic, but the Shiga toxin is pretty toxic. E. coli food poisoning leads to a dysentery. Both of these dysenteries by bacteria can be life-threatening unless you can get an IV to replace fluids, because these toxins break down the junctions in your colon, and so now you have interstitial fluid that floods out of your body faster than you can replace it. So the way you fight these two diseases is with IVs. You get to a hospital where they can put you on an IV and start administering antitoxin until the toxin is out of your system. All right, so that's the end of Chapter 9. Next time, I want to introduce you to a little bit of just a kind of a summary of biotechnology that's going on out there so that you're aware of it.